but not shortening, right? So maybe you're holding something up and not shortening the bicep muscle. Isotonic contraction, iso again, same or constant, tonic for tension. So this would be constant tension, but in this case we're going to keep that weight the same, and we can actually shorten or lengthen that muscle. Within this topic, we've got two sort of subtypes or subcategories to think about: concentric and eccentric, or eccentric, you can say it either way, contraction. Concentric would be shortening, eccentric or eccentric, lengthening, but you're still lifting some sort of weight that remains constant. And then our final word definition on the bottom is twitch, and that would be that mechanical response, so the contraction, right? That is the response to one action potential. All right. So, here we go. Here we've got some experiment set up for us. And they've set up a single muscle fiber with a motor neuron attached to it, I guess you could say. And on the top, they have a setup where they can stimulate that motor neuron, and that motor neuron can release its acetylcholine onto that single muscle fiber, and it will contract, right? And here they're measuring that tension that's generated during that contraction. So over time, we have our single action potential happening on that dotted line, and then there's a little bit of a lag time, or latent period is the official word for that. A little bit of lag time while all of the events occur. Remember we talked about all of the events, releasing calcium, blah, 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 right? Energizing our myosin head, all that stuff. Okay, so all that stuff takes a little bit of time to happen. That happens during this latent period. And then once we get those cross bridges to attach, we start generating the tension, right? So this is on a graph showing us how that tension rises, right, as we generate more tension from that one action potential. And then it eventually tapers off, right, as we pump the calcium back into the sarcoplasmic reticulum, right, and we detach everything, right, all that stuff. So this is how all of those events would look if you were to graph all of that stuff over time. And then we've got a separate experimental setup. We still have a motor neuron, we still have a single muscle fiber. This time, they're going to allow that muscle fiber to shorten with the contraction, and here, they're measuring how much, or what is that distance that the muscle fiber actually shortened, right? Like maybe it shortened 1 centimeter or 2 centimeters, right? Something like that. So we have our single action potential happening again. We've got our little latent period while everything happens, calcium, blah, 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 right? Cross bridges, everything. And here we're measuring that distance, right, of the muscle fiber shortening. So as time goes on and as the tension increases, if we allow the muscle fiber to shorten, you see how it gets shorter, right? And then as the tension decreases, the muscle goes back to its original length. All right, so these are just ways of looking at the stuff that we know, right, can happen in graph format. All right, so those things, right? Let's say one twitch, right? One sort of generation of tension and possibly shortening as a result of one action potential. One of those things. We saw that that happened over a certain period of time, right? Because we were graphing over time. So different muscle fibers will twitch, taking up different lengths of time. And that depends on how do we build that individual fiber. We have different ways of building individual muscle fibers, turns out, right? So let's see how we can build different types of muscle fibers so that we can have muscles that have maybe different or variable built-in capabilities, depending on what we might want to do, right? Physically, what we might want to do. So, the two sort of things that we want to consider when we're building are different muscle fiber types. First one would be, remember the calcium ATPase that's embedded in that sarcoplasmic reticulum membrane, and its job is to pump the calcium back into the sarcoplasmic reticulum, remove it from the cytosol, right? Bring it back into sarcoplasmic reticulum, and we're going to start taking away, right, calcium so that we can end all of that contraction, right? So it turns out that we have a couple of different versions of that calcium ATPase, which we chose from our pie. Remember, we chose that from our pie. So we have maybe two different subcategories of that choice from our pie. We can have a fast-acting one, and we can have a slow-acting one. All right, so you just choose one from the pie, right? Either the fast-acting or the slow-acting calcium ATPase. All right, if we pick the slow-acting calcium ATPase, let's remember, right, the picture and what's happening with calcium ATPase. And if we have a slow one, what happens to the concentration of calcium in the cytosol if our calcium ATPase is slow? Say it louder. Yeah, it remains in the cytosol, right? We kind of still have a lot of calcium like hanging out in the cytosol, kind of for a long time, right? So if we still have a lot of calcium hanging out in the cytosol for kind of a long time, woohoo, what are we going to be doing? Like, what is the muscle going to be doing during that time? If calcium's still there. Contracting, right? The muscle fiber is going to keep contracting, right? As long as the calcium is there, as long as we have a lot of it, let's just keep contracting, right? Okay, so if we build that particular muscle fiber with a whole bunch of those, the slow calcium ATPases, all right, we can sustain long muscle contractions, right? Okay. So that's one thing. If we build that type of fiber, we're going to give it a name. We'll call it a slow twitch fiber. All right, that's good. So let's look at a different situation. Let's build instead, let's say maybe in a different muscle fiber, right? Let's use some fast-acting calcium ATPases. We'll embed that in the sarcoplasmic reticulum of that fiber. And what's going to happen to the calcium concentration in the cytosol when we build with those? Going to go down, right? Because we're going to be pumping the calcium back into the sarcoplasmic reticulum very quickly, and so we get rid of the calcium that was hanging out in the cytosol. So what's going to happen as far as contraction is concerned in that muscle fiber? It stops, right? And it stops sooner than that first muscle fiber with the slow calcium ATPase. So far, so good? All right, so we're going to give a name to that second type of fiber, we'll just call fast twitch. All right, that's pretty good. So far, I hope that that's like pretty straightforward. All right, so the other consideration that we can think about when we're building muscle fibers would be, how does that muscle fiber generate its ATP supply? And we already know we need the ATP for our big thing that we needed the ATP for was what? Like a big thing when we're contracting. What did we need ATP for? To do what? Yeah, it was something like the cross bridge part, right? And the myosin heads, right? We needed the ATP to detach the myosin head and then energize the myosin head, right? So that was our big thing for contraction that we needed ATP for. So, but what are our ways of generating ATP in any cell? Really, it doesn't even have to be just muscle, but in any cell, going back to metabolism. Oxidative phosphorylation, right? That was a biggie. And then our other one, not as much, but, glycolysis, right? All right, so those are our two choices, right? Our two big choices. And we know oxidative phosphorylation, we can generate a whole lot of ATP. Glycolysis, for one molecule of glucose, But if we had a lot of glucose and a lot of glycolytic enzymes, we can still, generate a good amount of ATP. All right, so those are our two choices. So depending on how we sort of tailor an individual muscle cell to make it more able to kick into oxidative phosphorylation or more able to kick into glycolysis, right? You see how we can use sort of either pathway, right? So if we wanted to build a muscle cell that would easily kick into using oxidative phosphorylation to generate its supply of ATP, what do we have to put in that muscle cell? Where do we undergo oxidative phosphorylation? Mitochondria, right? So we have to pack that cell with a lot of mitochondria, right? All right, and what did we need? Like what ingredient? Oxygen, right? Yeah, so if we need oxygen, we also have to have a good blood supply to that particular fiber. So because of that need for a blood supply, those fibers are highly vascularized and they tend to look more red because of that blood supply. So we sometimes will call them or refer to them as red fibers. But perhaps the more official name would be oxidative muscle fibers because that's the process that they will use as their first choice for generating their ATP supply. So let's look at a separate muscle fiber type that we're going to build. Let's say this separate muscle type, we want to rely on glycolysis, right? So where does glycolysis take place in any cell? Cytosol, right? Yeah, cytoplasmic cytosol. So, what do we have to pack these cells with in the cytoplasm? Enzymes, right? Specifically the glycolytic enzymes. We have to put a whole bunch of those in that cytosol, maybe more than in other cells, right? So that cell is ready, right? It's tailored to use glycolysis as its first choice for generating ATP. We don't have to have so many mitochondria in those, right? Because we're depending on glycolysis. So we don't have to have so many mitochondria. If we don't have so many mitochondria and we're depending on glycolysis, we don't need so much oxygen, right? So we don't have to have as good of a blood supply or as rich of a blood supply. So those fibers tend to look more pale compared to these red ones. So we could refer to those as white. But the more official name would be glycolytic, referring to they use glycolysis as their first choice, right, for generating their ATP supply. So glycolytic. So here we have our other two categories of how we can build our muscle cells, oxidative and glycolytic. So far, so good? All right, so now we've sort of built 4 choices, right, 4 ways of building the muscle cells. We're going to take these 4 ways of building and put them in different combinations, right? Because we could combine maybe this and this, right? Or this and this. You see that? Right? And then this and this and this and this, right? So let's see. It turns out in humans, we saw 4 possible choices in humans through evolution. We've kind of ended up with 3, right? 3 combinations that our muscles use. All right, so let's look at the combinations. At the top, slow, oxidative. So we've taken the slow calcium ATP ACEs and put those in the same cell that is going to depend on oxidative phosphorylation. So we've got a lot of mitochondria and a good blood supply to those. So we put those two ideas and those two structures, right, in the same cell, and we can combine the names, and we come up with one name, slow oxidative. All right, so far so good. Let's hang on to that idea and skip down to the bottom. Maybe we can have an opposite situation. We can pick fast calcium ATPases and put those into the same cell as the cell that's going to depend on glycolysis, right? So we don't have so many mitochondria, but we're going to have a lot of glycolytic enzymes put right into that cytosol. And that cell, okay, we have all that stuff in that cell. We'll combine the names and we'll say fast glycolytic. All right, so let's compare sort of the results from building these two types. So let's look at the top type, slow oxidative. So we've got a situation where we're letting the calcium hang around, right? And we're using a lot of oxygen for oxidative phosphorylation to generate our ATP, right? So, what is our result as far as like contraction goes, right? And tension generation and all of that. What do you think, right? We could do with that type of cell. We've got a lot of calcium hanging around, using up oxygen to generate our ATP. Huh? No, we're still in skeletal, so cardiac is a whole separate story. Oh my goodness. I laugh nervously. So just think of skeletal. Let's think of different kinds of exercises, right? So let's say our choices are, I'll just narrow it down to two choices. The choices are the Olympic bodybuilder who does, you know, that one with the big weight, with those big weights. And it's this guy, right? And he does this, and he does this, right? And he's like, and then he goes, right? That guy. Or the marathon runner, right? So those are our two choices. So which one do you think that top one? You think marathon. So let's talk about why. Why do you think it would be the marathon runner for the top one? Yeah. Good. So here we can sustain a contraction for a long time because of the slow, right? And marathon runners, they run for a long time, right? So they can sustain that tension in their muscles, right? For a long time. Okay, good. That's pretty good. We also know that marathon runners, obviously, they're breathing, right? A lot that whole time that they're running that marathon. I hope so. Right? My goodness. All right, so that also fits in to that idea. All right, so let's talk about this bottom one. I guess we're left with the weightlifting guy, right? Who's lifting and then, right? That. So I guess we're left with that example. Does that seem to fit in? What do you think? Like he's lifting this incredibly, incredibly heavy weight, right? And he lifts it for however many seconds he can, right? And then, right? This big sound, right? That guy. Does that seem to make sense with this one? Right? He can only do that for a few seconds, but it's a huge amount of weight, right? Huge. And then, but once he's done, right? And it's only a few seconds. So that kind of fits in here. We only have, because we're using the fast calcium ATPases, we only have that calcium available, right, for that contraction for a relatively short amount of time. And once it's over, right? You've got to drop that weight. Make sense? But these muscles can generate an incredible amount of tension. So that guy can lift a lot of weight, right? But only for a short time. All right, so we've got that. And then we've got a type kind of somewhere in the middle. We're going to pick fast, but we're going to use both processes. So we're going to build that cell with the fast calcium ATPases, but we're going to provide a good amount of mitochondria and also a good amount of glycolytic enzymes. So kind of depending on the situation, right? We could kind of go either way with our pathways for that one. All All right. Here's a slide showing just a cross-section through some muscle, showing us, right, red and white, and then our intermediate types of fibers. All right. And we can graph how much tension we can generate for how long for each muscle fiber type. So here we see slow, oxidative, right? So we can generate some tension and hang on to that, keep going, right, for a long time. And here we had our opposite, fast glycolytic, so we can generate a whole lot of tension, but only for a short time, and then it just, right, that drops. And then here in between, we generate, and then it'll kind of generate a bunch of tension. Oh, okay, that's pretty good. We can kind of sustain that, but you know, eventually, yeah, we're going to run out of steam. All right, so here we've got a summary table of all the stuff we've talked about, like I usually say. If this helps you, good. If not, go with the pictures and the explanations. Okay, so this graph basically tells us, it's probably more complicated looking than it really is. This is basically telling us that, you see how these three different lines are labeled: heavy load, light load, and intermediate load. We know that load is weight, right? So if we're lifting something heavy, this is pretty intuitive, actually. This is just looking at what we already know on a graph. But if we try to lift something heavy, right? Oh man, and this is distant, the distance shortened. So, oh man, we might only be able to pick that up so far, and then that's it for me, right? Forget it. So we can only shorten the muscle like this much. As opposed to a lighter load, oh yeah, see? That would be pretty easy. We could shorten the muscle a lot, right? A greater distance. And we also know that the steepness of a curve, when we're graphing against time, the steepness of a curve is how fast something is happening. So a steeper curve is faster, right? So same thing with a heavy load. Oh man, it's kind of slower, right? To shorten the muscle. Whereas if you have a light load, oh, that's quick. Good. So it's just graphing something that we actually already know. All right. So here's a more complicated graph. We're kind of building up to a very complicated graph. So here we've got a different looking graph, but still talking about stuff that we know, right, intuitively. So let's take a look at this one. Here we've got load. So instead of time, we've got weight, right, on this x-axis. And then here on the y-axis, we've got that shortening velocity, how quickly, right, we can shorten. And above 0 is shortening. Below 0 is lengthening. So we have to look, right? Whenever you see a graph, especially on the exam, right? Whenever you see a graph, look at all of these, right? The x and y axes, and even if it goes, like in this example, below, right? Below the x-axis, you want to really look at those to see and remind yourself, what am I dealing with here, right? All right, so let's look at this point, which happens to be along this line that is our weight, right? So here we've got, like, let's say 0 pounds, just as an example, right? And here we would be having like 1 pound, 2 pound, 3 pounds, right? On and on and on. So here we've got however many pounds that is, right? At this point, we're at 0, so we are neither shortening nor lengthening, right? We've got a contraction happening because we put the weight, let's say, in our hand, and we've got a contraction happening, but we're not shortening and we're not lengthening. Good? So we're kind of here with that. All right. So from here, let's go ahead and lighten the load, right? Lighten the load, so we're going to move this way, right? This is lightening the load. And as we lighten the load, we already know, right? We already know that we could shorten the muscle, and the lighter the load gets, the quicker we're able to shorten the muscle. All right, so that's how that idea would look. And when we get to 0 load, like let's say we put it down, but we still want to shorten the muscle, that's when we could shorten the muscle the fastest, when we're actually holding nothing, right? No weight. All right, so that's what this curve is going to look like. Let's look at the opposite direction. So now we're going to increase the load. We're going to put something heavier, right, in my hand. And as it gets heavier and heavier, that muscle is going to lengthen, or it can lengthen, right? Lengthen, lengthen, lengthen. And the heavier it gets, kind of the faster it's going to lengthen, right, if you allow it to lengthen. So far, so good? All right, so that's what this is going to look like. We should kind of recognize these words down here. What was isotonic again? Isotonic. Constant. Our choices were length or tension. Tension, right? So we'll kind of try to remember. Tonic starts with a T, and tension starts with a T. So maybe that's a way to remember. And metric is like a meter, right? And this is length, maybe something like that. So constant tension. So that's keeping the amount of cross bridge, right? Tension the same, and lengthening and shortening because the load is changing, right? That weight is changing. All right. So, oh no, we're almost at the very complicated graph. Let's look here at an additional idea about tension. Here we've got an action potential. So S stands for stimulus, or action potential in that muscle fiber. And when we've got an action potential, we already know we can measure a twitch, right? The tension generated in the contraction. Good. So this would be one action potential, one twitch. Great. Over time, let's have multiple action potentials, and we'll kind of space them out, right? So that they occur just as the previous twitch is kind of coming down, but it's not totally over, right? And we see, they seem to kind of sum, right? You kind of add them up. However, because the next action potential is happening when the twitch is kind of ending, we do have a little bit of kind of relaxation. You see that? We almost fully relax the muscle. We almost fully relax the muscle. So we're going to give this situation a name. First of all, we're going to use the word tetanus, which most people think of tetanus as, you step on the rusty nail, you're going to get a tetanus shot, right? So now we're going to have a new definition, right, of tetanus, which is combining these twitches or this tension, right? Because now we've got multiple twitches. So we'll call that tetanus. That's our physiology definition of tetanus. And this type of tetanus is unfused because we have some tension and then we relax a little bit, and then tension and we relax a little bit. We don't come all the way back down to zero, but we do get a little bit of relaxation in between each peak there. New situation. Let's put all of our action potentials really, really, really close together, right? When we do that, wow, we really add up all of those twitches and they become huge. We can generate a lot of tension and we don't allow that muscle to relax, right? So we'll still use the word tetanus, but we'll call it fused because we're just adding this all together and we get no relaxation in between the peaks. We're just always at this peak, and the peak is huge. Make sense? So this is when you're wanting to do something, right? And maybe you have to lift something and do that for a long time kind of thing, right? You want to keep the contraction going. That's going to be our fused tetanus.
Yeah, I was going to kind of ask the same thing. The unfused tetanus would be the reps with the weight. It kind of relaxes when you put it down. And
sort of. It could be. However, I don't want you to automatically assume that, let's say let's say these would correspond to 1 rep, 2nd rep, 3rd rep if we're doing these, right? I don't want you to assume that because it could be this is 1 rep if I just like made that weight higher, right? And I said, right? This could be, that could be 1 rep. And then we let it go. Woo! And then, right? All right, good. So let's see. This is a complicated slide that we've been building up to with all these graphs. So here's our most complex graph of the entire lecture, and I really want you guys to spend time over the weekend wrapping your heads around the graph, memorizing the graph, right? Everything in it, but also, of course, the ideas behind what is this graph trying to tell us. Because, I mean, not only for the exam, of course, right? Exam on Tuesday. But we're going to take this graph and we're going to have to use it in the next section when we talk about cardiac muscle. So that's why you really want to get this one to make cardiac somehow understandable. All right, so let's see what we've got. All right, let's start. Let's just set up the graph. Along our x-axis, we've got muscle length and we've got percentage of muscle length. So we're putting 100% not out here as like our maximum, but we're actually putting that in the middle of the graph. Kind of mathematically, that sounds like, wait a second, 100% is supposed to be like it, right? Sort of, technically. All right, but here when we're thinking about these skeletal muscles, right, we know that, okay, yeah, you could have, let's say, your what you would think at first as your 100%, like that's your maximum length. Let's say that's the muscle at rest. Let's say I'm going to rest, right? This bicep muscle. So that would be 100%. Sort of what we originally thought. And then if we contract and let the muscle shorten, that would be everything or anything less than 100%, right? If this is bicep muscle length at rest, and then I contract the muscle and allow it to shorten, all of these shorter lengths would be 80%, 60%, 50%, right? Of 100% of the original length, right? However, in muscle, we know that we can stretch, right? We can stretch skeletal muscles, and we had that titin, right? Remember that protein built in, the coil-shaped protein built in to our sarcomeres to allow for that, to allow us to pull those thick and thin filaments kind of away from each other as opposed to this way, pull them away from each other and still be able to, when we let go of that stretch, recoil, right? All right, so it's this stretch beyond that resting length. That is where this 120, 140, 160 comes in, right? So let's see, we can't really do it with elbow because this elbow is not going to go. But let's think of quads, right? I think quads work really well. So we have like a straight knee, right? And if I take this foot and I want to stretch my quad, right? If you guys were to do this, this would feel stretched out, right? That would be these lengths over here, greater than 100%, because this just kind of standing, not even standing, I'm going to stand on this leg, but I'm going to talk about this one, right? Maybe something like this, where I'm not actually using that quad, right? But it's kind of relaxed and not stretched either. Let's say this is 100%, right? Let's say when I stand on it, I might shorten it, right? But then if I pull, I could lengthen. All right, so, That's just the x-axis. All right. Let's see. What do we got on the y-axis? All right, we've got isometric, tetanic. That's tetanus, right? The I guess adjective version of the word tetanus, and tension. So, do we remember what that was? So, tetanus, right, was adding together all of our twitches. Okay, so we know that. And then isometric, constant length. Okay, constant length. Okay, so at a constant length, right? So we're generating some tension, but we're not shortening or lengthening yet, right? Whatever that might be, right? We've got a maximum tension that we can create. So you guys do that now, right? So you can feel this one, because I think this concept's harder than this one. So just focus on your bicep, right? And contract the bicep without, shortening your arm. So we're going to leave the arm extended, leave that elbow extended, and just contract the bicep, right? But leave this length the same. Good? So, and now contract it as hard as you can without shortening. Right? Woo! Awesome! Little personal trainer's action in there. Okay, good. So, that maximum, we're going to say that that's 100%. Make sense? Of an isometric tetanus. Make sense? All right. So, anything less than that as far as tension goes, so now we go from maximum, and we let go a little bit. Maybe that's 60%, right, if I let go a little bit. But the length doesn't change, right? You guys can do that too. Let go from maximum and just relax it a little bit. See how that's less? Still a little bit you can have, but less. All right. So, oh my gosh, this we've got on the y-axis. Let's look at this green line. And what are we really graphing here? I want to look right at the center. So right at the center is this 100% muscle length. So that was kind of here. We could think of that maybe as our starting point for this muscle, the starting length for our muscle. So we've got that, and this dotted line is hitting right at that point, right? That's where we're going to start. So it looks like at this starting length, right? It looks like, according to this green line, what they've graphed, right, out of measurements, experiments, right? It looks like we are capable of generating that maximum tension, right? That maximum isometric tension. Oh. All right, and let's look, they kind of gave us a little, I guess that's kind of like a key, right? A little reminder of this is what the sarcomere would probably look like at that length, right? All right, so we see thin and thick filaments. We've got, some overlap, right? And this is our starting point. All right, from here we can generate maximum amount of tension possible, which we already felt. Okay, that's pretty good. And a little bit above that, a little bit below that, we can still generate maximum. Awesome. Let's see. If we shorten, right? If we shorten the muscle, and we see here's our key. You see this is shorter? Right? We've got more overlap. If we shorten the muscle and we say, you know what, now this is going to be our starting point, right? So instead of, let's say you're going to go do a workout, right? And we'll stick with the bicep. Let's say now I'm going to start from here and I'm going to do my curl, right? I'm going to do that. Let's say instead of starting here, I decided to start here instead, right? Oh, I know, this is even better if you've ever done this. Let's say you're on the machine, so not even like free weights, but let's say you're on one of the machines. And the bicep machine, you get to rest your elbows on that thing. I don't know if anybody's ever known who's been on the machine for the bicep curl. Not like free weights, because it's very obvious when you're on the machine, this idea. So whoever's been on the machine and you get to rest, right? Have you ever noticed that if you start here, it's really easy for you to like initiate that curl. But if you start here, it's kind of like, maybe it's like you got to get over this hump, right, of effort if you start here. So you're always like to psych yourself up, like, right? Start here, right? So this is this idea. If you start your effort, right, with a shortened, like an already shortened muscle, you're only capable of generating like 80% of your maximum that you're capable of. Make sense? And if we start even shorter, like we're already shortened, right? We're only capable of generating, it's getting worse, right? And if we're like really shortened, oh man, eventually, if you really, really, really shorten the muscle, right here, we're going below 60% of the original length, right? Oh, brother, looks like we can't even generate any tension. This is our starting point, right? All right. Let's go in the opposite direction. Now we're going to lengthen, right, beyond 100%. Same idea, and here's our little key, right? Now we're pulling those filaments away from each other. We're lengthening that sarcomere. Same idea, the longer the muscle gets, the more we stretch it out, the harder it's going to be, oh my gosh, to overcome that extra length and generate anything near our maximum that we can generate, right, that we're capable of. And we will definitely hit a maximum length where we can't generate any tension, right? This, beyond about 170, 175% of that starting point, or 100%, that would be the equivalent of a muscle tear, right? You actually just rip the muscle, right? And we know if you totally rip a muscle, that's not going to work, right? You generate no tension because you've torn the sarcomeres. All right, so that is what this green line is all about. Telling us, okay, at different starting lengths for that muscle, there is a particular length where our muscle is the best at generating its maximum capability of tension, right? And then if you go too far in any direction on either side, you become less capable of generating your maximum. You still generate, right? Just not maximum. All right, so that is that. Let's see. Let's see, we talked about this. That's pretty good. I want to save. No, we can still talk about this. Let's talk about I was going to say I wanted to save the red, but let's talk about the red. Let's talk about the red. Oh, and here's this other key. I forgot to tell you this other key, right? So here, green, active tension. Here, red, passive tension. So active tension is action potential coming from motor neuron. If it's coming from motor neuron, we told that muscle, right? Up in cortex, right? Primary motor cortex. We told that muscle contract. We sent a signal down the motor neuron, acetylcholine, boom, right? So that would be active tension. Here for passive tension, this is more like, let's say, because I think the stretch part is more obvious. Let's say you now sit down on the floor and you're going to stretch, let's say your hamstrings. Or you do a bicep stretch, right? You can just hang on and do a bicep stretch, right? Like this, and you're just passively, not actively lengthening that muscle, right? as we do that and stretch, this type of tension is a very, very different type of tension. It has to do with those titin proteins. And because they're shaped like a coil, you know how any spring, right, there's tension in there and you kind of pull on the coil and the tension increases, right? And then you can kind of release that. That's what this is about, right? So we're not having any sort of action potentials, nothing like that, graded to nothing, right? EPPs, no. None of that stuff. This is all about the titin. Good? And we're kind of pulling on that coil. Good? All right. So, let's see. Aha! All right. So we've talked about ATP and generating ATP from glycolysis and or oxidative phosphorylation, right? And there's our ATP. And here we see, oh yeah, there's that myosin, right? And there's that calcium, ATPases. So we see that stuff. We know all of this stuff. And we know all this stuff. You can use amino acids, you can use fatty acids, or we know all this stuff. Glycogen and glucose, we know all that stuff. Great. Let's look at what is new in this situation. New in this situation. It seems like we've got another source for our ATP. Huh. Another source for our ATP. What is this? Creatine phosphate. Why is this coming at us, right? Out of left field. That is unfair. Right? Looks like we can take whatever this is. I don't know. And, take ADP and phosphate. Oh my goodness, and we're going to generate some ATP. Oh, great. And we're left with this creatine. Oh, why? Why is there a new thing? Unfair, right? Let's see. All right. New thing. New thing to memorize. Oops, I got cut off a little bit at the top there. But the title of this slide is Generating ATP for Skeletal Muscle. In addition to the ways that we already know, right? Okay, let's see, what is this creatine anyway? Comes from an amino acid, and it's made by the muscle cell. So it's generated, right, in the muscle cell. And creatine phosphate is that plus a phosphate, right? So phosphorylated version of that. We already know that putting a phosphate onto something, usually, especially if you're adding it onto kind of a negative area, that's a high energy bond, right? So this creatine phosphate is this high energy version of this creatine that the muscle generated, right, from an amino acid. Okay, so we've got this high energy thing, and we saw in the previous slide that we can take that phosphate group, right? Let's look at it. Take that phosphate group from that creatine phosphate, see there, and ka-chung, generate some ATP, and then we've got our creatine over here. All right, so we can do that. Okay, let's go back to this slide. All right, so it turns out that when you're starting a muscle contraction, especially if it's like, all right, time to start the workout, right? Like, remember when we used to do our jumping jacks? And I made you go for a minute, right? So let's say it's kind of during that time, it's like the beginning of the jumping jacks. But we're going to go for a while, right? Okay, at the beginning, right? The beginning of the workout, the beginning of a contraction, right? It turns out that we kind of, in any particular muscle cell, really only have a little bit of ATP kind of on hand at the beginning. We can always generate more, we know that, right? Glycolysis and for oxidative phosphorylation. Good? So we know that we can do that if we keep going, right? But at the beginning, all right, we have a little bit of ATP on hand, and well, that's going to be used up if we only have a little bit at the beginning, right? And we can use that up very quickly. All right. So, we have this backup plan. This whole creatine and creatine phosphate bit in there is sort of like our backup plan to generate some extra ATP in that little window, right? That little window between. Oh, I just used up that little ATP that was there, but oh my gosh, she's making me go. Like I have to keep going, right? And glycolysis and oxidative phosphorylation have yet to fully kick in and generate their ATPs. So you see the little window in there? Okay, so this quick, as you see that this, where it says number 1, you see how that's quick. It's just a one-step thing, right? We know glycolysis. How many steps? Oxidative phosphorylation, right? Rather. I mean, it's going to take a long time, right? Quick. So we can quickly just generate enough to get us to here, right? Woo. All right. So I put in here a little bit about this being the theory behind all those creatine supplements, right? You go to like GNC, right, and like, woo, and the athletes, right, taking these supplements to build up that creatine phosphate, right, so that in theory they have a whole bunch of extra of that, right, so that that little time period in there, they've got a supply, right? A good supply of creatine and the creatine phosphate. Make sense? All right, so that's the theory behind that. I kind of always ask, has anybody ever, I don't want me to put you on the spot, but has anybody ever tried these supplements or know anybody who's tried such supplements and any results? Like I always just ask because there's plenty of like people who swear by it and people who are like, Yeah, a lot of, yeah, exactly.
You know, a lot of people say that you just build water weight.
Yeah.
You know, and that can maybe be because you are creating more like mitochondria in your cells too, so you could kind of make an argument somehow for it. But I mean, you definitely have more energy. And,
Okay.
so I just don't know if it's studies have shown that statistically it reduces injury by.
Oh, see, that's interesting. All right. All right. So we have, like, as far as this little mechanism, we're not sure, but at studies saying reducing injury. I mean, that's kind of an interesting little side thing, right? That, I don't know.
Yeah.
Maybe that wasn't the intention, right?
It used to be really taboo also, and then also studies have shown that it really doesn't damage your liver the way. Initially, people would say you have to cycle it, or else you get damage, and the more studies have been shown, it's shown that it's not as harmful as we initially thought, or at least the taboo was that it was harmful. But
yeah. So liver things. Whenever we can maybe talk about this more when we get to talking about the liver, but anything, right? Any drug, whether it's like prescription or over the counter or illicit, including supplements, right? Including supplements, whether they say this is like a synthetic supplement or herbal or, you know what I mean, all natural or whatever. You always have to consider side effects. Liver and kidneys are going to be a big deal for clearing stuff, and that's a whole big thing. We'll talk about that more when we get to liver and kidneys. Wait, who had something?
What happens with the extra creatine in the body?
Oh, okay, so this is the liver part. This is the liver part, and also the kidney part, which we will talk about. We will talk about when we get to those.
It's just like excreting what.
Huh?
It's just excreting what leftover you don't need.
Exactly. So, having said that, having touched on that, we will revisit this topic when we get to liver a little bit, kidneys a lot, and so keep this. I mean, obviously for the exam, we're going to have that up front, but don't let it like, right? Because we're gonna come back to that and talk about getting rid of stuff. Good, like extra. If you take a supplement now, maybe you have extra stuff. All right. Did you have a question also? Oh, just okay, that's cool. All right. So, oh my gosh, let's see. Let's add a little bit more to the discussion. And here we've got some stuff that we actually already know from metabolism. We just talked about with this creatine phosphate thing, the very, very beginnings, right, of a contraction and let's say your workout, right? And how do we generate that little bit of extra ATP that's in that window, right? When we, use up what we had hanging around in the muscle, but yet glycolysis and oxidative phosphorylation have not fully kicked in that muscle. So we already talked about that. Let's say we're gonna keep going with our workout, right? All right, now we're gonna go, I made you guys go for a minute, right? But let's say I made you go 5 minutes, right? 10 minutes, somewhere in there with the jumping jacks, right? Let's say I did that. And jumping jacks, I would put that in the moderate category, right? That's not too bad. You know, but it's not, you know, just like little lame, right? It's not that either. So moderate. We already know glucose is our first choice to use. That's floating around the bloodstream. And then when we use that up, which is going to happen pretty quickly, we're going to tap into what? After we use up glucose. Huh? Not the fatty acids yet. Not proteins yet. Glycogen, right? So first we use up glucose that's floating around in the bloodstream, right? And then glycogen, right? That's second choice, right? Second choice. So we tap into glycogen. And where is glycogen stored? We had two big locations. Liver and skeletal muscle. This is our topic, right? All right. So, all right. Let's keep going. 5 minutes, 10 minutes. We've used up our glucose. We're tapping into glycogen, right? We tap into the liver stores, and we are working the skeletal muscles. Of course, we're tapping into the skeletal muscle stores. It's right there, right? Perfect. Isn't that smart, right? To have the glycogen stores in the place where you're going to use it if you keep going for 5 or 10 minutes, right? And you just used up all your glucose. Smart, smart. All right, so, all right, I'm pushing it to 10. We're going to that 10-minute mark, right? Oops, now we're going to use up all that glycogen in the muscle and the liver. Uh-oh, what's our next choice? Ideally, fatty acids, right? Ideally, our next choice, especially if our goal is losing fat, right? If we're doing the workout and we're trying to lose some fat, right? Our goal, ideally, is to use up all of those glycogen stores so that the body will kick into drawing upon our fatty acid stores. Make sense? All right. Then we burn the fat, right? That's the whole thing behind burn the fat. Fat burning, right? That's it. That's it. So, pushing, long workout, and hopefully we're tapping into, right, fatty acids. There's your big, long aerobic workout. Good? Super awesome. Let's see. This was assuming that the exercise was kind of moderate, like I said, the aerobic exercise, really do. Because we know those fatty acids, what was a great place to use those? It was a great place to like take off 2 chunks at a time, and then we could generate like, whoa, so many ATPs.
Oxidative phosphorylation.
Yeah, oxidative phosphorylation was like 139, right? Like ATPs for like 1. Awesome, right? So, woo, totally is tying together, right? If we're going to do like a half an hour, or let's say the whole hour aerobics class now, right? Or you go for a run, long run, right? Or whatever is your activity, right? Long, breathing, oxygen, oxidative phosphorylation, fatty acids. Good. It's all coming together, I hope. All right. Let's look at a different situation. Let's say, oh man, this is super intense exercise. We're going to go nuts now. Super intense exercise. And this is the type of exercise where perhaps not a lot of that oxygen, I mean, we're still breathing, not a lot of that oxygen is getting to the muscles. So we're going to be focusing now on which muscle fiber type. If we're going to, we're going to do a different type of exercise. And maybe we're done with the aerobics class, or maybe we're alternating days, or whatever. So now we're going to, let's do the weight. Let's have a weight training day. It's that day. All right. And we're going to do heavy weights that day, right? Increase heavy weights that day. So we're going to put that in the category of intense exercise. So let's do that. We're going to use glycolysis, right? So we're going to use our glycolytic fibers to do that. And let's see. We know from our knowledge of glycolysis, for one molecule of glucose, we don't generate very much ATP. However, if we had a lot of glucose and a lot of enzymes, right, glycolytic enzymes, which we do in the glycolytic fibers, well then we can generate a lot of ATP. We'd have to use up a lot of glucose, right? And we'd have to have a lot of enzymes available, but we could, right? So we already built our cells to have a lot of enzymes. All we have to do is supply it with a lot of glucose, right? All right. Well, we could use up a lot of glucose. We have our alternates, right? Glycogen, and we kind of mentioned fatty acids, but what was the other thing we had? You mentioned proteins, right? Proteins, and we haven't acknowledged them officially yet, right? We could fit proteins in there, right? All right, so that kind of fits in sort of over there. We also know our results, right, or one of our results from glycolysis was going to be lactate, right? If we were not going to take that pyruvate and continue through Krebs, et cetera, we were going to build up a lot of lactate, right? And we had that whole discussion about lactate. Well, now we're having that discussion again, right? Oh man, building up lactate. All right, so remember when we had the original discussion about lactate and we talked about muscle soreness, right? That delayed onset muscle soreness. All right, and we said that phenomenon, right? This delayed onset muscle soreness that days later, still sore, right? We said for a long time, many years, it was thought that it was due to that buildup of lactate. But then they did studies pretty recently and disproved that theory, and it was actually little kind of micro tears in the tissue, and it was just a sign of building, right? Building more muscle tissue. So it's still good, right? We're still building muscle. It's still good. But it didn't have anything to do with the lactate. But anyway, okay, that's that. That's the delayed onset muscle soreness. Setting that aside, right, as a separate thing. Let's just focus on the metabolism itself. We just made a whole bunch of lactate, right? I mean, period. There's no way around that. We did it, right? We just made a whole bunch of lactate. So, we got to do something with the lactate. You can't just let it sit there, right, forever. So we got to do something with it. We have to get it to go at least back to pyruvate, right, at some point in time. Let it go back to pyruvate, and then we can let it either continue, right, like through Krebs cycle and oxidative phosphorylation. We could do that. Or we could even backtrack from there. And we know we can do that, right, based on our knowledge of metabolism. Right? We can go back to pyruvate, and we can go further back in the process. All right, so we know that we can do those things. However, we need oxygen to do that. So this is why after you did your big anaerobic heavyweight training day, right, as an example, workout, right, and you built up all this lactate, right, after, like right after that workout, you know you're still breathing hard. This is why, right, because we need that oxygen to take any of that lactate and bring it back, right, to pyruvate, and we can either go up So we keep breathing to basically supply the lactate, right? With some oxygen to convert it back so that it's not hanging around too long. So we want to get as much of that as possible converted. All right. So there's that. So that kind of explains, right, why you kind of keep having to breathe, right, heavily. You don't just instantly. And we saw that. Remember when we were doing our respiratory rates, right? And I let you guys, like I had you guys measure your respiratory rate right after the exercise, and then we waited at a minute, right? And it kind of took that minute, right, to go from, oh, our respiratory rate was way up, and then it kind of came down gradually over the course of a minute. Probably we could have done like 30 seconds and then a minute, and then you would see how it comes down gradually. All right, so that kind of explains why that lingers a little bit. We've got to take care of this, this lactate. All right, we also have to use oxygen to replenish creatine phosphate, which we talked about before, right? Once we use that up, okay, finish out the workout, blah, blah, blah. We should rebuild that, right? For like next time that muscle is going to be asked to do something, right? So we should regenerate that. We also have to regenerate our glycogen supplies. I mean, we just did a big workout. We just used our glycogen supplies. So these things also require oxygen to do those. So again, continue breathing. We're going to give this idea a name. This idea of, oh, we have to keep breathing and keep supplying oxygen even after we're done with our workout, right? We're going to give that idea a name. We'll call it oxygen debt. And some people have heard of oxygen debt, like coming in to class, like especially if you work out a lot, people talk about the oxygen debt. And this is the oxygen debt, right? We kind of have to pay the body back in the form of oxygen. And the way we do that is to keep breathing heavily after the workout is over. And as these things are replenished, the breathing kind of quiets down. We're gonna have other factors entering into this process of going from high respiratory rate to low respiratory rate. We'll have other things factoring in besides this. And we will see those other factors when we talk about respiratory system and renal system. Incredibly complex, but we will revisit this idea. All right. So, let me see where we are. Hang on. Whoops. Okay, good. Ooh, let's see. Let's do this. Let's knock like this slide out of here. And then we can take our break. We've got tetanus, looks so confused, right? And then eventually that muscle just like gets tired, right, and gives up. We know that, right? Especially with that guy with the big weight, and right? So that's that. And then you've got a period of rest, and then we're going to try doing that again. You see how the second time around, right, with this much rest, oh man, can't maintain that like as long as that one, right? And we know that. That's kind of an intuitive thing. If we were to allow for a longer rest period, maybe we would have a better chance of sustaining a tetanus like the first time, right, if we had a longer rest. Good? But with a short rest, not so good. So this is part of the reasoning behind having alternating days, right, of similar exercises, right, especially if you're doing weight training. You want to alternate the days. This is part of the explanation there. Let's see. We've kind of talked about all this already, actually, but here you see kind of everything in all these words, like, so when you go back to studying, you can kind of read through. But basically, we've got all of our 3 types of fibers, and we will see in any one whole muscle a distribution of all three types, right? So you won't have one whole muscle, like a whole bicep, that has all of one type. We'll have a distribution of all three of the different types. That distribution, like let's say, you could have 30% of one, 30%, 30%, right? Because we've got the three types. So let's say you could have, or 33%, right, of each. You could have that in one person in, let's say, a bicep, right? Different person, if we look at their bicep, they could have a different distribution, right? Maybe 40% of one type and then 30%, 30%. So, right? Something like that. You could vary the percentages. That is genetically determined. Good? So you sort of inherit your basic distribution of the muscle types in your muscles. You will also have within one person different distributions in different muscles, right? But you can also go from person to person and have very, very different distributions within the same muscle. So we see different body types, and this is a lot of where these different body types, as far as muscle tissue is concerned, this is where the different body types come from, is the distribution of the muscle fibers. So we know, to kind of tie this whole discussion together, and then we'll go on our break, we know that there is a body type that happens to be the one that we seem to always see in the marathon runners, right? And there is a body type that we seem to always see in the big deal heavy weightlifters, right? Especially when you get to the elite levels. Like all the marathon runners seem to be these sort of wiry, lean, right? Long muscled and sort of smaller muscled builds, right? And the bodybuilder types tend to be, right, these sort of stockier, very bulky, right, muscle types. So a lot of that is based on how they were built, and that's based on DNA. So there is quite a bit of truth to that statement, you know, you're born with it, right? There's a lot of truth to that. You can always take whatever you're born with and work with it, right? And train and make yourself something. For someone who is born with the body type of, let's say, that lean, sort of long and lean and small muscled marathon runner, it will be easier for that person to train and become successful, right? The other person who's stockier and bulkier can still train to be a marathon runner. It's not like you're doomed, right? And you can never be a marathon runner if you're built like a bodybuilder. It'll just be harder, right? That activity will be harder for you. And if you want it, you'll have to train harder. That's the mental part. The mental part is very strong, right? Very strong. But you're going to have to work harder. And the reverse, right? If you're built like a bodybuilder, that's going to come naturally, right? It's going to be easier for you to train to get to a certain level. If you're a marathon runner type build, harder. You can do it, it's just going to be a lot more work for you to achieve that same goal. Make sense? All right. It's not impossible. Did you? Okay. Let's see. Big. Here we've got just, that got put up at the top, but just some vocabulary words. You guys can read through muscle atrophy and hypertrophy. Hypertrophy is building up muscle tissue. Atrophy is sort of withering away muscle tissue. And it basically has to do with disuse, like if you stop using muscle tissue, it's going to atrophy. And if you use muscle tissue, especially if you use it more, it will hypertrophy. So you guys can read through those vocabulary words. That's pretty straightforward. And here's just anatomy review, right, of agonist and antagonist and extension, flexion, all of that stuff. All right, so you can read through all those. So let's take our little break. After the break, we'll fly through a little bit of smooth muscle, cardiac muscle. That's super quick. And some muscle control, and then we'll do our lab. So let's come back at 9.